Welcome to the deep dive. Today we're venturing into the bustling universe within the human microbiome.
Right, that huge collection of microorganisms living in and on us.
And it's not just some, quirky biological fact. It's becoming really clear this microbial world is a major player in our overall health.
Definitely. And here's a mind-bending thought to kick us off.
Yeah.
The number of microbial genes in our bodies just dwarfs our own human genes. Think about the sheer untapped potential there.
Wow. Okay, so our mission today is basically to pull out the most crucial insights from all the research out there on the human microbiome.
Exactly. We've waded through a ton of information to bring you the highlights. The journey of discovery, who lives there, what they do, their links to health, disease.
Consider this your shortcut, your rapid immersion into a field that's, well, it's fundamentally changing how we see ourselves.
Yeah, absolutely. We've been looking at the sort of big picture of microbiome science.
From the very first glimpses.
To the cutting-edge tech revealing its secrets now.
Right, so let's jump in. How did this whole field even get started?
Okay, so our story really starts way back, 17th century, Anthony van Leeuwenhoek.
Ah, the microscope guy.
The microscope guy, yeah. He was the first to actually see these tiny animalcules, as he called them, in his own mouth even.
Amazing.
Then fast forward a bit to 1853, Joseph Leidy, his work, of flora and fauna within living animals. That's often seen as the formal beginning of actually studying the microbes inside us.
Okay, so those were the early glimpses. But then came the 19th century and the germ theory, right?
Yeah.
Pasteur, Koch.
Right. Huge figures. And their work, quite rightly, focused on identifying the specific microbes that cause disease, the pathogens.
The bad guys.
Exactly. Koch even laid out his famous postulates, the rules for proving a microbe caused a specific disease.
So the focus was really on disease back then.
Very much so.
Good.
But a really important shift started in the early 20th century with Elie Metchnikoff.
Okay, what did he propose?
He had this revolutionary idea for the time, that some bacteria, like the ones in yogurt, could actually be beneficial, maybe even help you live longer.
Wow, that's early for thinking about good bacteria.
It really was a very early hint that, not all microbes are out to get us.
And did that idea catch on?
Well, it gained some real traction with the isolation of Escherichia coli Nissl, 1917. You might even recognize that name.
Yeah, it's still used in probiotics today.
Exactly. So the idea of using specific bacteria therapeutically, that was recognized quite early on, like finding a helpful little worker in this microbial city.
It's interesting, though, because at the same time, the germ theory was dominant.
Right, but microbial ecology was also sort of bubbling up in the late 19th century, thanks to people like Bajorink and Winogradsky.
What were they looking at?
The bigger picture, really. How microbes interact in their natural environments, not just as disease agents.
Okay, so different streams of thought developing. Then, moving into the mid-20th century, what were the next big steps?
A really critical technical hurdle was figuring out how to grow anaerobic organisms.
The ones that don't need oxygen.
Precisely. Which, as we now know, make up the vast majority of microbes in our gut. Robert Hungate's work here was absolutely instrumental.
So learning to talk to the hidden majority, basically.
You could put it that way, yeah. And speaking of the gut, 1958, that's when the first modern use of fecal microbiota transplantation, or FMT, happened in Western medicine.
FMT, what was it used for?
Eisman and his colleagues used it to treat pseudomembranous colitis, a really nasty gut infection. Successfully, too.
Wow. Was that a totally new idea?
Well, interestingly, no. There are records from ancient Chinese medicine describing similar practices, using fecal matter for severe digestive problems centuries ago.
That's fascinating. Ancient wisdom meets modern medicine.
It really is. Sometimes these things circle back in surprising ways.
But the real, explosion in understanding must have come with molecular biology, right?
Oh, absolutely. The molecular revolution was key. Think PCR in 1983.
Polymerase chain reaction, yeah.
Right. And then the boom in DNA sequencing technologies. Sanger sequencing in the late 70s, then next-generation stuff around 2005, third gen hot on its heels in 2008.
So suddenly you could read the genetic code directly.
Exactly. Without needing to grow the microbes in a lab dish. That was huge. Culture-independent studies became possible. It was like getting the instruction manuals for this whole hidden world.
And who figured out how to sort of organize all that genetic information?
A pivotal step was Carl Woese's work back in the 60s and 70s. He focused on 16S RNA genes.
The microbial barcode, right?
That's a great way to put it. It gave us a universal marker to identify and classify microbes, mapping out who was actually there.
Then I remember hearing about the holobiont concept. When did that come about?
That emerged in the early 90s. It really pushed this idea that we aren't just individuals.
We're ecosystems.
Yeah, interconnected ecosystems, the host and its microbes functioning as a single biological unit, a grand symbiotic partnership.
Okay. And connecting that to the tech side, metagenomics, that sounds important.
Monumental leap. Metagenomics lets scientists analyze all the genetic material from all the microbes in a sample, all at once.
So not just identifying them, but seeing their collective potential.
Exactly. Understanding the entire library of this microbial city, not just a few books.
Which leads us, I guess, to the Human Microbiome Project.
Right, the HMP, launched by the NIH in 2007. A massive undertaking.
What was its main goal?
The first phase, HMP1, was really about mapping things out, characterizing the microbial communities in healthy adults across different body sites, gut, skin, mouth, etc., like a global census of our internal inhabitants.
And then there was a follow-up.
Yes, the Integrative Human Microbiome Project, or IHMP, that ran from about 2014 to 2016. It took things further.
How so?
It looked specifically at the microbiome's role in certain health conditions. They focused on 3 areas: pregnancy and preterm birth, the onset of inflammatory bowel disease, IBD, and the onset of type 2 diabetes.
And they generated a lot of data.
An unbelievable amount. Reference genomes, metagenomic data, all made publicly available, which has just fueled so much more research since then.
Okay, so what were some of the really big takeaways from the HMP overall?
Some really fundamental insights came out. First, each of us has a uniquely personal collection of microbes. It's like a fingerprint.
Really? So my microbiome is different from yours?
Significantly, yes.
Yeah.
Second, the sheer number of microbial genes we carry vastly outweighs our own human genes.
You talked about that, the potential.
Exactly. And third, these communities aren't static. They're dynamic. They change based on things like our diet, our lifestyle, medications.
So it's constantly shifting. Okay, this might be a good point to clarify terms. We hear microbiota and microbiome. What's the difference exactly?
Good question. Technically, microbiota refers to the microbes themselves, the actual bacteria, fungi, viruses, archaea, protists.
The living organisms.
Right. Microbiome is broader. It includes the microbes plus their genes and the environment they live in. However, in common usage, and even in a lot of scientific literature, human microbiome is often used as shorthand for the whole collection, including the collective microbial genomes, the metagenome.
So the terms get used a bit interchangeably sometimes.
They do. While there's a precise distinction, people often use microbiome to refer to that whole internal microbial world. It's just something to be aware of.
Got it. And you listed off quite a few types of microbes there. It's not just bacteria then?
Not at all. That's a really important point. Bacteria get a lot of the attention, but it's a whole diverse ecosystem. We've got viruses.
Including phages, right? Viruses that infect bacteria.
Exactly, bacteriophages. Then there are fungi yeasts, molds, archaea, which are single-celled but genetically quite distinct from bacteria, and even protists.
Wow.
And what's really exciting now is the growing focus on the dark matter of the microbiota.
Dark matter, like in space.
It's sort of analogous. It refers to the non-bacterial components, especially viruses and other microscopic things, whose roles we're really just starting to figure out. It's like exploring uncharted territory.
And these different microbes, they don't just hang out anywhere, right? They have preferred locations.
Absolutely. The body's a varied landscape. The gut, particularly the large intestine, is the big metropolis, most dense, most diverse. Trillions of microbes live there.
Okay.
But we also have significant communities on our skin, in our mouth, our nasal passages, lungs, the whole digestive tract, urogenital tract.
Each with its own specific crew.
Pretty much. Each site has its own unique conditions, nutrients, pH, oxygen levels, immune activity, and that shapes the microbial community that thrives there.
And the sheer number. I've heard estimates that microbial cells outnumber our own human cells.
The estimates vary, but yeah, it's thought to be roughly equal or perhaps even more microbial cells than human cells. It's a staggering amount.
Though some parts of the body are just generally sterile, right? Like blood.
Yes. In a healthy person, internal bodily fluids like blood should generally be sterile.
Okay. So we've established there's this vast, diverse community living with us, but why should you, listening right now, really care? What are these microbes actually doing for us?
Right, let's get into their functions. This is crucial. One of the biggest roles, especially for the gut microbes, is helping us digest food and get nutrients.
How do they do that?
They have enzymes that we lack. They can break down complex carbohydrates. Think certain types of fiber that our own digestive system just can't handle.
And what happens when they break those down?
Fermentation. This process produces short-chain fatty acids, SCFAs. The main ones are acetate, propionate, and butyrate.
SCFAs, are those important?
Hugely important. They aren't just waste products. Butyrate, for example, is a primary energy source for the cells lining our colon. They help keep the gut barrier strong.
So they literally feed our gut lining.
Yes, think of it like a VIP fuel, keeping that barrier healthy, preventing leaks. Plus, SCFAs have wider effects. They can modulate the immune system too.
That's amazing. So they help us extract more energy and nutrients from food. You also mentioned they make vitamins.
Yes, indeed. Certain gut microbes are little vitamin factories. They synthesize essential vitamins we need, like various B vitamins, B12, folate, biotin, riboflavin, and also vitamin K.
So they're filling in nutritional gaps for us.
Exactly. It shows how we're really co-evolved. They complement our own biology.
Okay, and immunity. You mentioned SCFAs influence it, but the microbiome's role in the immune system is bigger than that, right?
Oh, it's fundamental. Right from birth, our microbiome helps train our developing immune system.
Train it how?
To tell the difference between friend and foe, between harmless resident microbes, the commensals, and dangerous invaders, the pathogens. This education is crucial.
And the gut is a key place for this.
It's a major hub. There's a huge amount of immune tissue associated with the gut-associated lymphoid tissue, or GALT. The microbiome plays a big role in its development and function.
So they help educate the immune system. Do they also help defend us?
Yes, in several ways. They compete with potential pathogens for space and nutrients competitive exclusion. Plus, many resident microbes actually produce antimicrobial compounds that can inhibit or kill harmful bacteria.
Like their own little weapons system?
Kind of.
Yeah.
And going back to those SCFAs, they also have anti-inflammatory effects and, as we said, help maintain gut barrier integrity. A strong barrier prevents unwanted stuff from crossing into our system and triggering inflammation.
So they're like a training academy, a border patrol, and they have anti-inflammatory properties. That's quite a portfolio.
It really is. They're deeply integrated into our immune defenses.
Okay, what about metabolism? How do gut microbes affect things like weight and energy balance?
The gut microbiota acts almost like an extra metabolic organ. It processes things from our diet, producing a whole host of bioactive molecules that influence our energy homeostasis.
Like what kind of molecules?
Well, SCFAs again, but also things like secondary bile acids. These microbial products get absorbed into our bloodstream and can signal to various organs, affecting how we store fat, how sensitive we are to insulin, and so on.
And this can link to obesity.
There's definitely a connection being explored. The efficiency with which someone's microbiome extracts calories from their food might play a role. Some studies suggest differences in microbial makeup could make someone more prone to gaining weight. It's like having a more or less efficient internal fuel converter.
Fascinating. Okay, and the gut-brain axis. This one always blows my mind. How can bacteria in my gut talk to my brain?
It's a complex two-way street, a communication superhighway, really. It involves nerves, hormones, the immune system, and those microbial metabolites we've been talking about.
Some multiple channels.
Exactly. And get this, gut microbes can actually produce neurotransmitters themselves.
Like serotonin,
Yes.
dopamine.
Things like serotonin, GABA, dopamine. Or they can influence our body's production of them. These are the chemical messengers the brain uses. So by influencing neurotransmitter levels, microbes can potentially affect mood, behavior, even cognitive function.
Wow.
And when this gut environment gets disrupted, a state we call dysbiosis, it's being increasingly linked to neurological and psychiatric conditions.
Such as?
Things like anxiety, depression, even neurodevelopmental conditions like autism spectrum disorder and neurodegenerative diseases like Parkinson's and Alzheimer's. The links are still being intensely researched, but the connection is becoming clearer.
It really underscores how interconnected everything is. So this dysbiosis, this imbalance, that seems to be a key factor when things go wrong.
Precisely. Dysbiosis is strongly implicated in a whole range of diseases, particularly autoimmune disorders.
Like IBD?
Yes, inflammatory bowel disease, Crohn's, ulcerative colitis, but also rheumatoid arthritis, type 1 diabetes, multiple sclerosis, lupus.
What are the common patterns seen in these conditions?
Often, there's a reduction in overall bacterial diversity, fewer types of microbes. And specific shifts, certain groups might increase while others decrease. For instance, some animal studies in lupus point towards an increase in Enterococcus gallinarum, and other studies using genetic approaches have suggested links between certain bifidobacterium species and increased risk for type 1 diabetes or celiac disease.
It's complex, though. Correlation versus causation.
Always the challenge in this field. but the associations are strong and consistent across many studies.
Okay. And we touched on obesity and metabolic syndrome earlier. What's the latest thinking there?
Right. Obesity often correlates with lower gut microbial diversity. There's also talk about the ratio of two major bacterial groups, firmicutes and bacteroidetes, though that's perhaps a bit simplistic now.
But there's experimental evidence, too.
Yes, the animal studies involving fecal transplants are quite compelling. Transferring the microbiome from obese mice can make lean mice gain weight, suggesting a causal link. And dietary changes that help with weight loss often improve the microbiome profile, too.
Makes sense. What about type 2 diabetes? Is there a clear microbial signature there?
Again, growing evidence points that way. People with type 2 diabetes often show altered gut microbiome composition. For example, some studies find higher levels of bacteria like Prevotella copri.
And how might that link to the disease?
Well, microbial metabolites are thought to play a role again, as CFAs, but also things like branched-chain amino acids, BCAAs. Plus, dysbiosis often goes hand in hand with low-grade systemic inflammation, which is a known factor in developing insulin resistance and problems with glucose metabolism.
Okay. We mentioned the gut-brain axis. What about other organ connections, gut-liver?
Yes, the gut-liver axis is critical, especially in liver diseases like MASLD, metabolic dysfunction-associated steatotic liver disease. That used to be called non-alcoholic fatty liver disease, NAFLD.
How does the gut affect the liver in MASLD?
The thinking is that gut dysbiosis can lead to a leaky gut, increased intestinal permeability. This allows microbial products, like bits of bacteria or toxins, to leak out of the gut and travel to the liver via the portal vein.
And that causes problems in the liver.
It triggers inflammation and contributes to the fat accumulation and damage in the liver. Specific microbial changes are often seen too, like more fusobacteria or fewer Ruminocaceae, along with changes in microbial metabolites.
It really seems like the gut microbiome's influence reaches almost everywhere. Back to the brain for a second, you mentioned autism, Alzheimer's, Parkinson's, depression.
Right. The gut-brain axis is implicated in all of those. The potential mechanisms involve disruptions in neurotransmitter production, increased inflammation crossing the blood-brain barrier, and altered immune responses, all possibly influenced by the state of the gut microbiome. It's a very active area of research.
It's incredibly complex. So, given all this complexity, how do scientists actually study this hidden world? What are the main tools they use?
Well, a cornerstone technology is metagenomics.
We mentioned that earlier, sequencing all the genes.
Exactly. It's a culture-independent approach. You take a sample, say, stool, extract all the DNA, and sequence everything. This gives you a picture of the entire genetic potential of the community.
What microbes are there and what they could potentially do?
Precisely. Shotgun metagenomics gives you high resolution, but it generates massive amounts of data that require powerful bioinformatics tools to analyze.
Okay. And what about 16S RNA sequencing? We hear that term a lot, too.
Right. 16S sequencing targets just one specific gene, the 16S ribosomal RNA gene. It's present in all bacteria and archaea, but has regions that vary between different species.
So it's like a barcode for bacteria.
That's the idea. By sequencing this gene, you get a good overview of which bacteria and archaea are present and in what relative abundance. It's generally cheaper and computationally less demanding than shotgun metagenomics.
But it has limitations.
Yeah, the resolution isn't as high. Sometimes you can only identify down to the genus level, not the species or strain. And crucially, it doesn't directly tell you about the functional capabilities, just who's there.
So metagenomics for potential functions, 16S for a census. What about actually growing the microbes? Is culturing still relevant?
Oh, absolutely. There's been a revival actually called culturomics.
Culture.
Yeah, it uses a huge variety of different culture conditions, different nutrients, oxygen levels, etc., combined with rapid identification methods like MALDI-TOF mass spectrometry.
To try and grow things that were previously considered unculturable.
Exactly. Because sequencing tells you what genes are there, but to really understand what a specific microbe does, its physiology, its biochemistry, you often need to grow it in the lab and study it directly. Culturing is essential for functional studies.
So genomics gives the blueprint. Culturing lets you see the machine running.
That's a good analogy. And ideally, you combine approaches.
Which leads to multi-omics.
Right. For the most complete picture, researchers try to integrate data from multiple omics layers. So genomics, metagenomics, the DNA, transcriptomics, metatranscriptomics, which genes are actually being turned on the RNA, proteomics, metaproteomics, which proteins are being made, and metabolomics, which small molecules or metabolites are being produced.
Wow, layers upon layers.
It gives you a much more holistic view of what the microbiome was doing dynamically and how it's interacting with the host, putting all the pieces of a very complex puzzle together.
It really sounds like we're, I don't know, just scratching the surface still. What are the big frontiers? Where is microbiome science heading?
There are so many exciting directions. One major push is for deeper functional insights. Not just knowing which species are there, but which strains, and understanding their specific molecular mechanisms. We're learning that different strains of the same species can act very differently.
So getting much more specific.
Exactly. And personalized medicine is a huge goal. Imagine diagnostics or treatments tailored to your unique microbiome.
That would be incredible.
Research is also expanding way beyond the gut. The skin microbiome, the lung microbiome. Understanding their roles in health and disease is a rapidly growing area.
And the influence of diet and lifestyle seems key.
Absolutely. How things like fiber, fermented foods, exercise, stress, how they shape our microbiome is a massive area of investigation. Plus, there's growing interest in the microbiome's role in aging and age-related conditions.
You mentioned the integrative HMP looked at specific diseases.
Yes, the IHMP provided really valuable longitudinal data tracking people over time related to type 2 diabetes risk, IBD onset, and pregnancy preterm birth.
Right.
It generated these rich multi-omics data sets showing dynamic changes, like microbiome instability before IBD flares, or distinct patterns in people developing prediabetes.
Okay, so we're learning more about disease connections. What about treatments? We mentioned FMT.
Right, FMT is remarkably effective for recurrent Clostridioides difficile infection, a really tough gut infection. That's its main established use.
Is it being explored for other things?
Oh, yes. Research is ongoing for IBD, metabolic syndrome, even some neurological conditions, but the results are more variable there. It's not a magic bullet for everything.
And besides FMT?
Well, there's continued interest in probiotics, live beneficial microbes, prebiotics, food for beneficial microbes, and symbiotics, a combination. And a newer area is live biotherapeutics.
What are those?
These are essentially specific, well-characterized live microorganisms developed and regulated like drugs to treat or prevent a disease. It's a more targeted approach than, say, traditional probiotics or FMT.
So moving towards more precision, but it can't be easy translating all this research into actual therapies people can use, right?
Yeah.
What are the hurdles?
You're absolutely right. There are significant challenges. The sheer complexity we've talked about, host-microbe interactions, are intricate. Then there's the huge variation between individuals. What works for one person might not work for another.
Standardization seems like it would be an issue, too.
Big time. We need standardized methods for sample collection, sequencing, data analysis, so we can compare results across studies reliably. Establishing clear cause and effect, not just correlation, is always difficult, and we need robust regulatory frameworks for things like FMT and live biotherapeutics.
And more large-scale studies.
Definitely. Large, long-term studies that track people over time using these standardized multi-omics approaches are critical to really nail down mechanisms and test interventions properly.
Okay, so as we wrap up this deep dive, it's been a fascinating journey through this inner world. What are the main things you hope listeners take away?
Well, I think the key points are the microbiome is this vast, diverse community within us. It's not just sitting there. It plays fundamental roles in digestion, immunity, metabolism, even brain function via the gut-brain axis.
And imbalances, dysbiosis, are linked to a growing list of diseases.
Exactly. And while the science is complex, we have these powerful new tools, genomics, multi-omics, giving us incredible insights. The future really holds huge potential for personalized approaches to health, leveraging our understanding of these microbial partners.
It really does make you think differently about yourself, doesn't it? Considering this profound impact, it leaves you wondering, what are the subtle, everyday things I'm doing that might be influencing this hidden world inside me?
Right. What are the downstream consequences, maybe years later, for health and well-being?
It's definitely worth pondering your own relationship with your microbiome, maybe exploring those dietary and lifestyle factors that we know can help shape it for the better.
Absolutely.
Well, thanks for guiding us through that incredible complexity, and thanks to all of you for joining us on this deep dive.